Hope is a belief in the probability of the possible rather than the necessity of the probable. A very good morning to one and all present here. My special greetings to our honorable guest and speaker of today's lecture, Dr. K. Thangaraj, CSIR, Bhatnagar Fellow, Center for the Cellular and Molecular Biology, CCMB, Hyderabad. Professor Manjayya, DH, Dean of Faculty of Science and Technology, Mangalore University, who will preside over this inaugural ceremony. Senior Professor and our Chairman, K.S. Sripada, the Department of Applied Zoology, and Professor M.S. Mustaq, the Convener, Department of Applied Zoology, and Dr. N. Narasimhaiah from the Department of Applied Zoology. I extend my heartiest welcome to you and request you, dear dignitaries, to come on to the dais and take your respective places. Let us applaud them with a clap. Dear esteemed guests, professors and students from various departments, we are delighted to invite you to a special lecture on the compelling topic of human origin, health and disease. Join us as we delve into the importance of integrating genomics into our understanding of the population structure, migration patterns, and the evolution of various traits. Let us now officially begin this session as I invite my friends to sing the university anthem. Thank you, dear friends. The soul should always stand ajar, ready to welcome the ecstatic experience, says Emily Dickinson. Respected distinguished guests of the day, we are truly honored to have you with us today. Your presence enriches this gathering, and we are excited to share this special occasion with such a distinguished group. Here comes Professor M.S. Mustaq, the convener from the Department of Applied Zoology, to extend cordial welcome to all of you and introduce the guests to the gathering.
Most respected Professor Manjaya, Dean, Faculty in Science and Technology, presents of today's program, esteemed guest of honor, Dr. K. Thangaraj, CSIR Bhatnagar Fellow, and Senior Professor Sripada Chairman, Dr. Nasimaya, my friend, and esteemed colleagues, research scholars, beloved participants, dear students, supporting staff members, friends from media, universities, ladies and gentlemen. I extend very warm and cordial welcome to one and all present here to special lecture on human origin, health and disease, organized by the Department of Applied Zoology. It is honored to welcome today's guest speaker, Dr. K. Thangaraj. And to talk about him, it is too big, so no time, that's why I am shortening his biodata. Dr. K. Thangaraj started his career as a scientist B at CSIR CCMB, Hyderabad. K. Thangaraj is presently a CSIR Bhatnagar Fellow, is the most precious award at the Center for CSIR Center for Cellular Molecular Biology, Hyderabad. He was director of the Center for DNA, Fingerprinting and Diagnostics, CDFT, Hyderabad during 2019 and 2023. He was also director additional charge of Institute of Stem Cell Science and Regenerative Medicine in STEM, Bangalore during 2022 to August 2022. His main research interest of the last 3 decades has been the field of population and medical genomics, and he has published about 300 research articles. He is elected fellow of Indian National Science Academy, Indian Academy of Science, National Academy of Science. He is a recipient of several awards including J.C. Bose Fellowship, Sun Pharma Research Award in Medical Science, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Scientist Award, Sir C.V. Raman Memorial Lecture Award, Sir Dr. U.N. Brahmachari Award, and delivered several orations. He is a board editor of Mitochondrion, associate editor of PMC, Medical Genomics, Tropical Medicine and International Health, and editorial board member of Human Genetics and Clinical Genetics. He was a president of Indian Society of Human Genetics during 2011 to 2015. He is the founder and president of Society of Mitochondrial Research and Medicine. Among all, he is a mentor for me, and we are very collaborating since 2007. We have co-authored many papers and many more to come. So many of his publications are freely downloaded from his institute web page. It is my proud to say that late President Dr. APJ Abdul Kalam quoted his work in his speech, notably while inaugurating the Malabar Institute of Model Science in Kerala. So it is a pleasure to have you with us. Hopefully this is the second visit. I think during 2016 he had come, and certainly not the last visit to our department and the university. We look forward to fruitful collaboration in future as well. I kindly request Professor Manjaya Dean to thoroughly welcome Dr. Thangaraj. Professor Manjaya presently serving as a Dean, Science and Faculty, and is a senior faculty and senior professor in the department. Dr. Manjaya, distinguished senior professor and chairman in the Department of PG Studies and Research in Computer Science at Mangalore University, where he also served as a Dean of Science and Technology. Over 30 years of experience in teaching and research, Dr. Manjaya has made significant contribution to the field of advanced network computing, mobile ad hoc networks, digital forensics. He holds PhD in Advanced Network Computing program with a focus on quality services and performance analysis, and MTech from National Institute of Technology, Karnataka, Surat. Mr. Manjaya has guided 22 PhD scholars, many of whom are now working in prestigious organizations, education institutions, both media and abroad, both in India and abroad. Currently, he is supervising 10 research scholars, including 2 from overseas. His expertise include routing algorithms for mobile ad hoc networks, vertical handoff mechanism for 4G networks, and the development of digital forensic laboratories in India. A prolific researcher, Dr. Manjay has authored and published around 167 papers in reputed international national journals and conferences. He has delivered over 235 invited and keynote talks at various international conferences, symposium, workshop, training sessions globally. His work has garnered over 1,077 citations with H index of 17 and I-10 index of 27. Dr. Manjaya has successfully managed large-scale research projects funded by agencies such as UGC, ESDSR, VGST. He is currently principal investigator and director for prestigious 20 crore project under PM Usha scheme by Ministry of Education, Government of India, aimed at enhancing research and academic activities in Mangalore University. We are honored to have such accomplished academician and researchers with us today. Please, I request our chairman, Professor Sripada, to thoroughly welcome our team. I would like to welcome our Professor Sripada, Chairman. He is the backbone of our program and he helps and supports all the programs in the department. I request Dr. Narasimha to thoroughly welcome Chairman. I also welcome Dr. Narasimha, faculty in Department of Applied Zoology. I request our chairman to thoroughly welcome Dr. Narasimha. I also welcome the faculties from different departments, neighboring departments from biotechnology, botany, and students from botany and bioscience, biotechnology, biochemistry, and all. So I would like to see it is a very enlightening talk, and please carefully listen and do think so much and ask many questions. Okay? Thank you. I welcome you once again. Thank you so much.
Thank you, sir, for your genuine words of welcome. It is my pleasure to introduce our esteemed speaker today, Dr. K. Tangaraj, who shared the 30 years of scientific contribution. We, Department of Applied Zoology, Mangalore University, would like to felicitate and provide the citation of his contribution. Therefore, I request our esteemed speaker today, Dr. K. Tangaraj, CSIR Patnagar Fellow, Center for Cellular and Molecular Biology, Hyderabad, a leading expert in the field of population genomics in India, to accept this gesture of appreciation. Now I request Chairman, Senior Professor of the Department of Applied Zoology, Professor Sripada, to read the citation.
Very good morning. It's a very rare occasion where all of us have joined to felicitate very rare personality. No? Yesterday, when Dr. Mushtaq came and suddenly told that the person who is having a lot of interest in science and having established collaboration with our department is visiting the neighboring institute, Neta Institute, and why not we invite and make an opportunity for our students to interact with the person. So interestingly, he agreed that even though he has got a talk in a molecular, he just saw that okay, because the students are very much interested to share his feelings. So organizing program, I welcome personally from my side, and also this rare occasion where I cannot read each and every aspect of his contribution for science. In citation, very highly appreciated and recognized contribution done by Dr. Rangaraj is being mentioned here. I just read the citation being given by the university by our department. Bangalore University, Mangalamurthy, in recognition of the outstanding contributions to the field of science, Dr. K. Rangaraj, CSIR Bhatnagar Fellow, CSIR Center for Cellular Molecular Biology, Hyderabad. Dr. Kumaraswamy Rangaraj is a distinguished scientist and a visionary leader in the fields of population and medical genomics. Dr. Rangaraj's remarkable contributions over the span of 3 decades have not only advanced our understanding of human genetics, but have also paved the way for groundbreaking discoveries that have far-reaching implications for public health and medicine. Currently, serving as a CSIR Bhatnagar Fellow at the CS Center for Cellular and Molecular Biology in Hyderabad, Dr. Rangaraj has an illustrious academic and research career highlighted by his tenure as the Director of the Center for DNA Fingerprinting and Diagnostics from 2019 to 2023, and his role as Director Additional Chair of the Institute for Stem Cell Science and Regenerative Medicine in 2020. Dr. Rangaraj's profile, output of approximately 300 research articles, showcase his commitment to scientific inquiry and excellence. His work has significantly deepened our understanding of population genetics, contributing valuable insights into the genetic diversity and health of population. As an elected fellow of the Indian National Science Academy, the Indian Academy of Sciences, and the National Academy of Sciences, he stands as a beacon of scientific integrity and innovation. His accolades, including the prestigious J.C. Bose Fellowship, the Sun Pharma Research Award in Medical Sciences, and Sir Dr. U.N. Brahmachari Award, just testifies to his outstanding contributions to science. Additionally, his leadership roles as President of the Indian Society of Human Genetics and founder of Society for Mitochondrial Research and Medicine demonstrate his unvarying dedication to advancing the field of genetics and fostering collaboration among scientists. In editorial capacity, Dr. Rangaraj has shared his expertise as a board editor of Mitochondria and associate editor of several STS journals, influencing the direction of research in medical genomics. His profound impact on the scientific community is marked by numerous orations and presentations, where he has inspired both current and future generation of researchers. Today, as we honor Dr. Kumaraswamy Rangaraj, we not only celebrate his scientific achievements, but also recognize his role as a mentor, leader, and advocate for the advancement of human health through genetics. His dedication to excellence and his visionary insights continue to inspire all of us. We deem it a great honor to felicitate him during a special lecture on human origin, health, and disease held on March 20, 2025. Dr. M.S. Mustar, the convener of the program, Department of Brain Zoology, Dr. K. Sripada, the chairman, Department of Brain Zoology, Dr. K. Raju J.S., Registrar, Mangalore University, Dr. K. Dharma, Honorable Vice Chancellor, Mangalore University. Thank you.
May I now request Dr. K. Tangaraj to address the audience with his introductory note.
The Dean, Chair of the Department of Applied Zoology, my friends, both Mustaq and sorry, faculty members of various departments, students. It's indeed my great pleasure to be here. Particularly in Mangalore. In fact, I went to Nita University, just informed that Mustaq that I am coming to Mangalore, and immediately asked me to come over here and talk to all of you. I'm very humble and it feels great to be honored by all of you. I didn't expect that such an honor is going to be. Humble and great to accept this honor. So any occasion comes like this, definitely it benefits the department and also the students, because not only me, when anybody comes to the university and delivers, they will bring their own experience which they acquired for a period of time, not in years, in decades. So what I advise you, that I will not talk much because anyway I am going to give a talk, just to listen the talk, keep it very simple for you, because I know that it's very diverse audience, for you to easily understand and how we in India, although with so much constraint, do the research. Right? So I basically come from a very good place, and you can see that how I could manage to do research. And even after my superannuation, because I retired a couple of years back, and now the organization, particularly the scientific organization, is supporting my research in the next 5 years, as you mentioned, got the Bhatnagar Fellowship that allows me to continue the research. If somebody put hard work, that actually pay a lot. So this is because of my hard work for the last 30 years. Now I can see that how it is helping us for understanding our Indian population, our history, and the problem, and how we can solve the problem. So that's what I'm going to speak, so I don't talk much about it. And just listen and see if you have any question, you can interrupt me, or you can ask towards the end of the talk. So once again, I am grateful to all of you for this great honor. And for the students, please listen and work hard. This is the time you all can spend a lot of time in reading and take inspiration from others and do extremely well for your future, either academic or research. Wish you all the best. Thank you.
Thank you, sir, for obliging to our request. Challenging the status quo takes commitment, courage, imagination, and above all, dedication to learning. We have amidst us Professor Manjaya PH, Dean of Faculty of Science and Technology, Mangalore University, who is committed and full of innovative ideas. I humbly request you, sir, to exchange your innovative ideas with the young minds gathered here.
Namaste.
Hariom, respected, a great personality, chief guest of this, one of the lecturing series from the Department of Applied Geology, the great Dr. K. Tangaraj, Bhatnagar Fellow of this country, Chairman, Senior Professor, Professor Sri Padani, and convener of this program, Professor M.S. Mustaq, Professor Dr. Narsim Mayati, respected faculties, and Mr. Chandru sir, who is the always part of our program, most of the activities, Mr. Chandrasekhar, my dear research scholar, and other faculties of the different departments, and dear creamy students. I stand before you with a great permission of Honorable Vice Chancellor Professor P.L. Dharmati, beloved Registrar Sri Raju Magavira, and the two other statutories, Professor Sangappa Devendra Padi. Friends, I take a quotable quote of great Swami Vivekananda mentioned, we need an education, we want an education by which strength of intellect is expanded, character is formed, by which one can stand on his own legs. Ladies and gentlemen, end of the education, and you have to identify what is our living things on this great globe between the explore as well as the blue sky. Such a wonderful personality, because of even though after his superannuation is come all the way. Please, it's my request with all of you, draw lines between a great root cause of Dr. K. Tangaraj and ourselves. You can find your own area of specialization. Such a wonderful area of specialization of the problems we can discuss after today as well as tomorrow and after tomorrow also. Continue. Friends, we don't like to disturb you because last 72 hours, correct, your mind, body and legs and hand become totally pain. But today, you have a break time and think about how to get through the insight and knowledge-based environment, because that is a wisdom. Without that one, impossible to recognize the society. That is the education. Please make sure yourself how best way you can going to utilize this lecture series, and you can go with your queries. And those who are interested, don't disturb others. Better you log out from this room. Because those who are very much interested to concentrate, let them enjoy, let them utilize, let them extract, they can survive easily. This is my request on behalf of the university. Once again, on behalf of the university, I personally extend a warm welcome to you, Professor, because you are such a friend. Getting fellowship is different. Such a wonderful scientific, especially Department of Science and Technology, Bhatnagar Fellow is not so easy. That is not like a behind the people, that is not the behind the whatever it may be, but only three T's. One is a discipline of the research, another one is a dedication, third one is a determination. These are the three aspects. But such a wonderful, simple, humble and jumble personality in front of us, it's my request, all of you, kindly utilize systematic manner. We have one and the other way. You can find yourself such a location where you can grow easily. Once again, congratulate the Department of Applied Geology, in which way they organize the program, which are going to be somewhat different manner, which are inside your knowledge, in addition to the scopus which are going to be take place for your syllabus, but additionally, how you are concentrating your area of specialization. Once again, each and one of you, make sure utilize properly, positively. You can go with your queries, questionnaires, and listen is very sensible manner. And wish you good luck, each one of you. Thank you, Ananda. Jai Hind.
Thank you, sir, for your benevolence and availing yourself to be with us for this session. The miracle of gratitude is that it shifts your perception to such an extent that it changes the world you see. With this in mind, I now request Dr. N. Narasimhaya to deliver the vote of thanks. Over to you, sir.
Respected dignitaries of the dais, it is a great pleasure and honor to propose a vote of thanks in this special lecture occasion. So, Dr. Thangarajan sir is our, I can say, Bhatnagar awardee. It is an Indian Nobel Award, I can say. Because as our president has said, the Bhatnagar achievement is a great achievement for as far as science field is concerned. So, Dr. Thangarajan sir, our convener and my friend, in 2016, we had in Mangalore University and joined hands with Applied Geology organized Science Congress. So that day we had a lot of scientific festival and also science we enjoyed. So second time we was visiting on behalf of Applied Geology personally, all in this occasion, all the insightful words and wisdom given, sir, personally on Department of Applied Geology and Mangalore University and also with the Karnataka, I special thank you for this special lecture for sir. Thank you. And as you listen, our team has given inspired all our youngsters. Despite his busy schedule, yes, taken as a president of this occasion, sir, on the Department of Applied Geology, and also personally, I extend a special heartfelt thanks to you, sir. So always encouragement is very significant for any event. So this is a very short program. We are busy with the tomorrow 2 days program. So emergency, our friend. So we know that forensic science and human science scientists are traveling to Mangalore last 3-4 days with our sister research institutes, Nitya. I hope that. And all the stalwarts are there, I think last 2-3 days before. So in this event, our the Thangarajan sir was arrived. So we discussed and for this, our chairman was supported lot immediately and action was initiated to organize this event. I extend very special thanks to our chairman, Professor Sripada, to given permission and support. Thank you. Taking lot of pain to organize this event, I special heartfelt thanks to my friend Professor Mustaq to organize all this event very significant manner. Thank you. And also I give a very special thanks to all young faculty and research scholars. Yesterday this hall was not in the very good look. They have made it some magic thing. So now, for all their effort, I extend very heartfelt thanks to them. And also, our wonderful audience. Without your engagement and involvement, no program will get successful in the university or anywhere. So I extend special thanks to all our young energetic minds of our students for their participation. I extend very special thanks to all the sister department. They have allowed all their students and also the faculty department chairman botany and biosciences, all the guest faculty and other faculties are given hands with us, joined. So I extend heartfelt thanks to all of you. Thank you.
So what I'm going to talk to you is about population history, particularly Indian population history, and how studying Indian population history is helping us for better understanding of our health and disease. So I must thank Professor Prasad for inviting me to talk to you. As I mentioned, was listening detail. So first let us see what is genome. All of you must have known. In simple term, genome is a complete genetic information present in the cell. There are two different kinds of genome. One comes from nuclear genome, one set of genomic information, and another one is from the mitochondria. I hope that you know that mitochondria has its own genome. While we inherit nuclear genome from both the parents, we have 46 chromosomes, 23 we get from the mother, 23 from the father. Right? Whereas we all inherit the mitochondrial genome only from the mother. There is no paternal contribution. Right? But you all know the human genome was sequenced, and Francis Collin from National Institute of Health, so he has his own way of defining the genome. Since genome is a short manual with an incredibly detailed blueprint for building every cell. He also says that it is a history book, a narrative of the journey of our species through time. And most importantly, he says, genome is a transformative textbook of medicine, with insights that will give healthcare providers new powers to treat, prevent, and cure the disease. So during my talk, you actually realize how important the statement of Francis Collin is. Now coming to the populations, as most of you are aware, just over the world have such a huge diversity, particularly human diversity. Right? Human diversity, look at, these are stratified in four major linguistic groups: Dravidians, Indo-European, Austroasiatic, and Tibetan. Right? These are four major linguistic groups. If you take Dravidians, there are four languages, you all know. Right? The southern states speak Tamil, Malayalam, Kannada, Telugu. Same way, there are several languages in Indo-Europe, several languages in Australasia, several languages in Tibetan. In addition to that, there are several isolated languages like Andamanis, Meghalis, and Nippon. Under each linguistic group or each language, there are several social groups, so-called caste system, upper caste, middle caste, lower caste, tribes, and primitive tribes. So all of this put together makes India is one of the complex nations. Let's switch on this light, these two lights. So now if you look at India, there are 4,635 anthropologically well-defined population groups. You don't see anywhere in the world such a huge diversity. Why studying diversity is very important? I wanted to show this particular slide. You know, Dubzhansky is one of the well-known evolutionary biologists. He wrote an article in the journal called the American Biology Teacher, where he has written the article, title of the article is, "Nothing in biology makes sense except in the light of evolution." So he realized how important was studying the evolution. Right? Again, you will understand towards the end of my talk how true this statement is. Right? Now let's look at how the human evolved, because we have to look at some evolutionary aspects. How many of you believe in evolution? Others don't believe in evolution. Or I can say, How many of you don't believe evolution? No one, right? That's true. So we have evolved human and chimpanzee from a common ancestor called hominids. It's not just one day we are turned into human. It took 7 billion years to evolve the way we look like today. During the 7 billion years, there are several species which have evolved and most of them have vanished. So you can see there are 7 million years. And during this course of evolution, there are some changes in our anatomy has happened. And for this, there's a genetic played a major role. You cannot actually separate genetics and evolution. Whatever the genetic change, that brings new features. That's the way the evolution takes place. In this process, there are 3 distinct features. Right? One of the distinct features is our ability to walk with 2 legs. Okay, we have evolved to walk with 2 legs when compared to chimpanzee or any non-human primates. That's one of the things. Second one, we gained ability to make tools to kill the animal hunting for food and so on. The third one is our brain size has increased. Look at the early species chimpanzees, the brain size was as similar as chimpanzee. That's increased to 35% in Australopithecus, increased to 50% of the modern human, 60%. And to our surprise, our close relative in terms of evolution, Neanderthals, it was predicted the brain size was larger than the human. But we don't know whether they are more intelligent than human. But this species is no more. Interestingly, one of the evolutionary biologists from Cambridge, he wrote in one of his articles, for the reason why the Neanderthals are not surviving today. He said the inherent violent behavior of a modern human probably responsible for vanishing of the species. I think you all agree, right? How violent we are, we know, right? So if think about the genetic change which brought the evolution, brought the modern human. There are very distinct mutations which helped. One of the things which I mentioned was the brain size, right? Mutation in myosin heavy chain gene actually helped the gorillas to create more space. Therefore, the brain size has increased. That's one. There are several other genes which are responsible for increasing brain size. Don't think that the gene is present only in human, myosin chain. Most of the genes are present in non-human primate like chimpanzee and other monkeys and so on. But the mutation in those genes is the one which brought the change in our human life. And I can tell you that even single base mutation, which can turn from male to female human life, one single base out of the million base, 3 million base. They are surviving. We have several such cases where X and Y is present, the individual developed as a male. But single base mutation, in a gene called androgen receptor gene, one base has turned the male phenotype into female. And that itself says that how the mutation actually changed our phenotype. There's a gene called buprenorphine that involved in learning, experience of pain, perception, behavior, memory. Is very, very important. Again, mutation in those genes. Because these are some of the unique features of human, right, when compared to chimpanzee. FOXP2 is another gene which is essential for speech and languages. Even if you don't believe on that, some of you, but I'll just take 3 examples. One example is, you see this, there are 3 brothers who born to a normal parents. The parents are absolutely fine. But you can see 3 brothers, they look like chimpanzees. That means there's a mutation which helped in evolving the way we look like today. There's a back mutation which again taking millions of years back, like features like this. This is one example. Second one, look at this child. One of the features which we gain through the evolution is lots of hairs on our body. Okay? But again, there's a back mutation, broad hairs everywhere on the body of this child. The third one, very recently was published, it's a Turkish family. I mentioned the ability to walk with 2 legs is part of the evolution. Right? There are 5 children, 1 son and 5 or 4 daughters in the family. Only the son can walk with 2 legs, but he can't walk steadily. He walks like a drunken person. But the 4 sisters, they can walk with only 4. They can't walk with 2. Because one of the features which we gain in the course of evolution, maybe a single mutation or clusters of mutation, which again took millions of years back. And this is where somebody wants to look at it. There's a YouTube video where you can see this Turkish family house. So all of this tells that genetics played a major role in the evolution. Without genetics, there's no evolution. Okay? That's one concept. Now I'm coming back to the Neanderthals, close relative of modern human. Very recently, a lot of studies have been done, and the skull, other bones have been found in Europe. Neanderthal Valley is the first place where this has been found. And very extensive work was done by Samantha Pavo. He was the first one to sequence things, several hundreds of thousands of year old bone, successfully sequenced and compared with modern bone. And that actually gave a kind of bridging the gap between modern human and other species which have shown earlier. And we know that because of his extensive work, he has been awarded Nobel Prize in 2022 in the field of physiology and medicine. It is 75. Right? But one can ask question, if we all evolve, and Neanderthal is very close to modern human, whether we all carry some amount of Neanderthal genome, right? You can ask, right? And one of our studies where large number of authors, and Pavo was also there, myself, and David Reach, one of my close friends and collaborators from Harvard. He studied 300 human genomes from different parts of the world and found that all the humans who are living outside Africa has up to 3% of Neanderthal. We all carry some proportion of Neanderthal. So this is direct link for the human evolution. So now I come back to the slide again. Now let's look at how human have evolved. The human evolution effect. The modern human evolved in Africa about 160,000 years back. But the question is, from Africa, how did they migrate and populate all over the world? There are 2 hypotheses. One suggests that the modern human took northern land route That is a connection. And went to north, and from there they spread. Others did not believe on that because the climatic conditions, even today in Europe, is very cold. Therefore, this may not be the right one. The alternate hypothesis is the southern coastal route. Okay. This is based on some of the archaeological evidence present in some of these places. So, we are working on various population in India for the last 3 decades. And to our surprise, we found that the first out of the human is in Indian subcontinent. Anybody knows who are they or who they could be? Any guess? Student? Okay, I now show the photo. Can you tell who are they? No? So they are the tribal populations of Andaman and Nicobar Islands. There are 4 tribes, because you are not aware that in Indian subcontinent, there are people typically like Africans or Levites. Right? There are 4 tribal groups: Ogi, Jawas, Sentinelese, and Great Almans. Their numbers are very small now. But they are in various islands. So that gave us opportunity to go and collect sample from them. Of course, it was more than 25 years back, just with the permission of Government of India and the local government. We had an opportunity to collect samples from Ponge, and there are a lot more behind that. I don't have time to tell that. And also, we had an opportunity to collect the bones which are kept in Dartmouth Museum in Cambridge. You may ask why these skulls are being kept in Dartmouth Museum. When the Britishers captured India, and when they wanted to construct the cellular jail, some of you may aware, as most of you may not aware about the cellular jail. This is Port Blair. Britishers constructed the cellular jail. There are 7 such wings to put all our freedom fighters. And each window is the back side of one of the cells. There are 7 cells. The back side and front very narrow. And front they cannot see our freedom fighters. They can see only the wall. Back side they cannot see because it is covered. That's the way our freedom fighters are being kept. We have to know some of the history also. So the reason why I am showing here is when they wanted to construct this jail, where the tribes were in the middle, therefore they have killed most of them. After killing, they took the skull. At that time, I don't know why they have took the skull. They took the skull and spread, but now there is a thing as to collect piece of bone, do the genomic analysis, compared with the present day Andaman tribes. So we used different kind of genome, and for this study we used complete mitochondrial genome. There are 16,500 base pairs, and this is circular genome. And based on the mutations present in the mitochondrial genome, we can assign a specific haplogroup. This is mitochondrial DNA, parsimonious tree, stepwise. Set of mutations defines the L haplogroup, which is predominant in Africa. After L, there are set of mutations define M or N. Then every step there are set of mutations. So I don't want to confuse you. Just see that every country or continent are predominant with a specific haplogroup. This is the way we try to construct and try to connect these. You can see African have different, India predominantly M haplogroup, Europe, you can see the Middle East different, Australia and India are different, North and South America are different. When we sequence the mitochondrial DNA of the Andamans, because they are features, one would expect they will fall in L, either L1, L2 or L3, L2. But to our surprise, they did not fall in any of the known haplogroup, but they acquired unique set of mutations because they are in isolation. You also should remember that certain part of the genome mutate frequently, certain mutate after tens of thousands of years. Based on this mutation, we can connect the people based on the haplogroup. Because they are in isolation, they acquired mutation. The numbers are nothing but 200 position, there is a mutation. Same way, 1524 position, there is a mutation. So these mutations are very unique in Andaman tribe, not there anywhere else in the world. Therefore, we assign the new haplogroup for the M31 and M32, and based on this is proposed that probably the Andaman tribe, or the first modern human, migrated out of Africa and taken southern coast and reached Andaman and Nicobar Island about 65,000 years ago. So this is the first genetic evidence suggesting that Andaman is the first modern human who migrated out of Africa. Right? Of course, this work was published 20 years back, and there's a lot of news about that everywhere, including BBC, National Geography, about this particular work, because the very first time it demonstrated that the Andamanis are the first modern human who migrated over us. The same issue of science, there's another study by Macaulay and Martin. They studied a population called Orang Asli from Malaysia. So they also come to the same kind of conclusion. Probably the southern group of migration is the possible way. And because of the mere coincidence, same issue, Peter Foster wrote an article called Perspective, highlighting both the studies, first study and study, and shown the route. So far there is no contradiction. This remains the same, that the first part of Africa migration took place taking the southern coast. Now I move on to another aspect of looking at the nuclear genome. After the human genome was sequenced using 1 million genetic variations, we thought of understanding the entire Indian population. So we used large number of population groups, population selected from all 4 microlinguistic groups, all social groups. Earlier I said that castes, various castes, tribes, primitive tribes, and so on. So in this study, we have shown that in the prehistoric India, there were 2 founding populations. One we call them as ancestral South India, other one is ancestral North India. Ancestral South Indians are part of this early human migration, because Andamanis and Africans are people with diverse. While the Andamanis migrated, taking southern coast to some groups stayed south, Kerala and Andaman. They are the ones called as ancestral South Indians. There was second wave of migration towards Africa, towards North. One group has come to Europe, other group has come to northern part of India, which we call as ancestral North. But still, there is a lot of genetic relationship between Europeans and ancestral North Indians. One example is the skin color mutation, the light skin color mutation. When everybody moved out of Africa, everybody should be like Andamans, right? But why some group are with the light skin color? Because of the evolutionary pressure. There's a mutation introduced because we need to synthesize vitamin D with the help of sunlight. In Europe, most often sunlight, even today, weeks and months they don't see the sunlight. You can imagine 50,000 years back. Because of the limited source of light, the melanin content is more when we are with the dark skin color. The mutation which turns skin color to lighter, the melanin content is reduced. Therefore, even with very mild light, like the light, it will be able to synthesize. So that's one example. There are many such examples which you have shown. I am not showing here. That's the way these two founding populations happen. And these two founding populations are mixed and gave rise to many populations. I'll come back quickly. Look at the other side. That shows the diversity of Indian population. These are all different Indian populations. These are European samples, African samples, and Chinese. There are hundreds of samples. Genetically, if different individuals, there are 100 individuals, are same, they form a type person because genetically they are the same. Whereas look at Indians, every single population is genetically unique. Therefore, they form a unique person. Although every single population is unique, but still we all share some kind of genetic. We are unique, but we all related genetically. So that's one point which I wanted to mention. I will come a bit later about that. So this shows that we are all unique, every population, but we are all connected. Shows that India is not inhabited by a single large population. Rather, you can say India is inhabited by large number of small populations. Small populations, we have 4,635 population groups. Each one is very unique. That the reason for that I come to you. I mentioned that these two groups are mixed and gave rise to many population groups. So I wanted to show you that when that had happened. Another study, it is large number of samples from north and large number of samples from south. And this shows that initially these two founding populations gave rise to many populations independently. For example, in the jungle, group of people are settled, and after finishing all the food material available there, some group must have migrated to other places. Okay. That's the way the migration of people taken place initially. But during the last 2 to 4,000 years, these two founding populations are mixed. The color shows that how the genome is admixed. After admixing, for the last 2,000 years, every single population started maintaining the endogamity. Endogamity means marrying into the group. So that's very common across country, right? Every population, the marriage happens very rarely now, the inter-caste or inter-religious marriage are happening. Otherwise, every single population, majority of them follow a endogamy marriage. So now, I will take you through the impact of this endogamy, marrying within the group for the last 2,000 years, how it has impacted on our health and disease. To understand that, now we wanted to go and explore not only within India, outside India, particularly South Asian groups. When you look at South Asian as a whole, there is much more diversity, 5,000 populations. Right? The reason why we wanted to go and look for other South Asian groups, there are a few examples, the reason for that. So one very, very important is that this is one of the studies where we have looked at a mutation in a gene called myosin binding protein C3. This is a gene, and there is a mutation. We found 25 days of deletion, which lead to a disease called cardiomyopathy. It's heart muscle disease. The mutation present on the both the copy of the chromosome, or the chromosome, the individual died at very early age. But the mutation exists in heterozygous condition. The individual survive up to 40-45 years, then it leads to sudden cardiac attacks. And more vulnerable are the athletes and sportspersons. The sportsperson carries the mutation and they are very active, then sometimes they collapse.
So that study actually gave us a lot of information that this mutation is well spread across South Asian countries, but not in other countries. So that shows that the mutation arose in India about 33,000 years back and spread within South Asian countries. So that's the reason. Let's look at the South Asian population. And we took 2,800 individuals from 275 enormous population. These are the place where we collected sample. All South Asian countries, including Sri Lanka, Pakistan, Nepal, Bangladesh, and other countries. So this study, to our surprise, although the study was taken for population aspect, then we found there's a possibility that because most of the population in India are having high IBD score, identity by descent score, therefore we predicted that most of the population in India are expected to have population-specific recessive mutation might lead to population-specific recessive disease. To give you in this IBD score, there are two populations we have included, Asana, British and Finnish population. Each line represents one population. There are 275 such lines are there. If the line is longer, that means the IBD score is higher. In simple way, you want to say the line is longer, IBD is more. That means individually in that particular community have come from or more related come from a common ancestor. For example, you are comparing 100 individuals. Out of 100, if 90 are having the same hepatitis, that means their sequence or maybe several is identical, you expect all of them to come from a common ancestor. If 90 are having a large such a DNA same, then they may fall here. If 50 are here, 10 are here, then there is no relatedness than it goes. That's the way one has to look at it. You can see this very tiny IBD compared to many of them, and they have large number of diseases have been well studied, and you can also see most of them are autosomal recessive. And you can also see Finnish population, large number of diseases. When you have such a tiny IBD, some large number of diseases, you can imagine what is the consequences of large number of population. Okay. So we just tell you that how does this manage. I told you it is a dominant. You can probably classify the disease into dominant or autosomal disease, autosomal dominant and autosomal recessive. We have 46 chromosomes, exists in 23 pairs. One chromosome of the pair we get from father, one from the mother. If one of the chromosomes carries a mutation, still it expresses a disease, that's a dominant. But the recessive mutation, if one chromosome carries a mutation, doesn't express any disease, that's a recessive. But that recessive will express the disease when the other chromosome, homologous chromosome, also has the same mutation. And it's called as homozygous. Right? Now look at this pedigree, how the mutation arise and spread into the community. For example, you know pedigree, right? Circle indicates female, square male, then this line indicates the marriage between them. Put line down, these two are their offspring. Both are fathers. So imagine this is a mutation exists in heterozygous condition, recessive mutation, therefore there is no disease. Then it goes to the next generation because her husband doesn't have this mutation, right? So it goes to the next generation. Again, both the daughters marry somebody from outside without mutation. There's no issue. So the mutation goes generation after the generation in a heterozygous condition. Same way on the other side goes generation after the generation in heterozygous condition. Now, let's come to the stage. In the same community, two individuals, now they say that they are unrelated. But actually, if you trace, before several generations, they are related. Right? Now they say that they are unrelated, but they are the same community, carry the mutation in heterozygous condition. There is a possibility that they marry and they contribute the abnormal chromosome to ninth generation. Therefore, there's a disease. So about 25% of chance this family are having the recessive disease. I'll give you a few examples. There's a gene called polymerase gamma, which is essential for mitochondrial DNA replication. There's a mutation that will lead to multiple mitochondrial DNA deletion. As you see here, the intact 99.9 kb pattern, but when there's a mutation, it introduces multiple mitochondrial DNA deletion. This is the wild type. Parents are heterozygous. C is the wild type. P is the mutant. Parents are heterozygous. You can see the electrogram. Hopefully you must have studied sequence, right? So the homozygous, you can see there are two copies of all the nucleotides. Both the chromosomes are identical. Here, in this particular position, in one chromosome carrying C, other chromosome carrying the mutant P. That's why in the same case, there's both C and T. That's called as heterozygous. Both the parents are heterozygous. Absolutely fine. No disease at all. After the marriage, you can see that it's unfortunate that P, 2 out of 3 children carrying the mutation homozygous, but they have several diseases, including encephalopathy, seizures, stroke-like episode, and most importantly, they die at a very early age. This child died. Here you can see 2 children have died because of homozygous. So that is very, very common among Indians. So now I go back to this. Of course, that is a different kind of study of mitochondrial disease when we study. Here we have so many populations with high values. So we selected a few populations. South India. We want to go back and see, because when we collect the sample, we don't have any idea about whether the population has the disease or not. But we went back, we found there is a high prevalence of junctional epidermis epidermolysis bullosa. So the disease severity you see, skin rashes, mucositis, and congenital heart disease. The children doesn't survive after 2 months of birth. And the reason is because they have 11 base pair deletion in a gene called LAMB3. So the first time we found there's a novel mutation, 11 base pair deletion, and we looked into few families and found this mutation exists in that population. Because it's a novel, we wanted to check initially our own database. Recently, probably you are aware that BBT sponsored a national program to sequence 10,000 human genome from 83 populations. Recently, that has been released by the Honorable Prime Minister, was online, and we all part of it as still coordinator of this program. We checked this 10,000 people. None of them carry this mutation. Right? Then we went on to check the global database of 8,62,000 individuals. These are all different databases. We found only 2 individuals with heterozygous human lady, but we could not find exactly the population name. But there are South Asian, but none of them carry this mutation. And if you look at chromosomes, it's more than 17 lakhs chromosomes. Right? None except those two. That means it's ultra rare mutation. Similarly, there are many such populations going to have some kind of population specific mutation. When the mutation is novel, no study at all, we have to understand whether this mutation is a real cause or by just coincidence we are finding. To understand that, what we need to do, we need to look at the impact of the mutation. When we look at the complete amino acid, the gene codes for 1172 amino acids. This is where the mutation. Because of the mutation, there is a truncation of amino acids become 799, removes approximately 380 amino acids. When the mutation truncate the protein, it becomes small protein, obviously cannot do this function, therefore there's a disease, right? We all understand that. But we need to demonstrate whether the phenotype is really because of this or not. So what we need to do? We need to create an animal model using CRISPR-Cas technology. You all must have studied. You can precisely predict or introduce a mutation and see the phenotype. We use mouse model just to remove that 11 base deletion, and you can see the pups. The pups doesn't survive beyond 2 days. The human children survive only for 2 months. The pups die within 2 days after the birth. But still you can see there are blisters on these mouse, pups of this mouse. But unfortunately we could not do any further experiment. Then we created another mouse model, because then we do CRISPR-Cas, sometimes it's an off target, it removes different size fragment. Fortunately we got 145 base per deletion, which includes this 11 base per deletion. Although the deletion is quite big, but this is the non-frame shape, but it removes only 15 amino acids. Therefore the impact is much lesser. So we allow the animal to grow. Up to 4 months, no problem at all. But after 4 months, it's slowly showing the symptoms as we see the child. Skin rashes. Right? Even the internal organs are affected. Then after 6 months, the animal shows the symptom typical to the child, and the animal dies, doesn't survive beyond 6 months. Then we dissected out and see the internal organs. The heart is enlarged, dilated. You can see a dilated parametric kind of phenotype. Spleen is enlarged. Stomach is strengthened, not because not eating much. Because we did the micro CT and so on, and before that I want to show the skin section. This is the white type. You can see there is an epidermis or very tightly packed, right? But in this animal, the particular case, you can see there is an epidermis, there is a gap. This is where the blister starts and shows those phenotypes. When we looked at the micro CT, this is small, the way we look at the CT of human, we have looked at the CT of lab case when they look at the pups. In wild type, heterozygous not much different. But in the homozygous, you can see the enlargement of heart, thickening of this intraventricular septum, but also there is a septal defect, there is a hole in the septum. All this leads to the septal related disease. Cross section also you can see the thickening of the septum and also other part of the heart. And there's a lot more work which I am not showing here because of time. Then, knowing all this, as a responsible scientist or responsible citizen, we need to do something for the society. We went back again because we found 11 base pair deletion, which is responsible for the disease, and we found mother was pregnant, and we wanted to do prenatal diagnosis. We brought the mother to Chennai, because this family is in extreme south, collected the amniotic fluid and tested just by simple PCR, and this is the homozygous, heterozygous, and the amniotic fluid showed 2 bands like heterozygous, not like right, the one. And we further confirmed by sequencing. As you see here, there's a overlapping peak, confirming that there's a 7 base pair deletion in the heterozygous condition. So we advise the mother to continue the pregnancy, and the child born about a year back, and with the proper phenotype and there's no issue at all. So at least this gives us some kind of satisfaction that the work which we started about 3 decades back, the population continues. Now we identify some population might have population specificities, going to them and looking at the disease prevalence and helping the family gives really kind of satisfaction. To extend that, to do any further work in human, we don't have homozygous mutations because children are dying. We took mother sample, heterozygous, then we did iPSC. We convert the iPSC into the homozygous deletion, as you see here, and we tested for its pluripotency using different markers, and now it's ready for converting that into different organs, including skin and also heart and so on, so that you can see the human cell how it is behaving. So that's one part which is going. Then we developed that as a characterocyte, the one which I showed earlier. This is the wild type, which is perfect, but with the homozygous mutation, you can see the skin structure is not proper, and also subsequently with the different, say with the different antibodies, the wild type is intact, but here is not intact, the human cells. Okay. So we are continuing with this work, this is very recent work. And finally, I wanted to show you that how we are taking it forward. We wanted now get approval from Tamil Nadu government, public health department, to screen the entire population. And we are almost at the final stage of getting approval from Tamil Nadu government. Once they give the permission, we go and screen. There are 5,000 individuals in that particular village, and the mutation is highly prevalent. Once you screen and identify the mutation, with the help of the local public health department, we start giving counseling, both pre-marital, those who have mutation. If, for example, both the boy and girl want to marry, carry the mutation heterozygous, then we advise either not to marry, or if you marry, go for prenatal diagnosis at every pregnancy. There is a chance. If one is heterozygous, one is wild type, no problem. So this way, if we continue with this, we can in fact able to eliminate this mutation in the long term. Therefore, the population can produce the normal children. It's not immediate kind of act, but it takes much more time. But that will help us in achieving that if we do it for multiple generations. In this line, when I was a director of CDFD, I introduced, because with this background, a new research program on pediatric rare disease. There are 16 institutes across India who are working on genetic diseases, not part of it. CDFD is the one which leads that. They screen for known mutations, don't find anything, then they send the sample to CDFD. CDFD will do either exome sequencing or the whole genome sequencing. And once they identify the mutation through initial check using different tools, if the mutations are really going to be pathogenic, then we use different animal models, cell line based, Drosophila zebrafish mouse, based on the conservation of the gene, and handling these two are much easier. Mouse, we don't find anything compatible here, then we use the mouse model. And we also try to treat some of the diseases, particularly intellectual disease. And most importantly, we have awareness programs. Somebody said you can just go to this special program on pediatric rare disease, and there are videos in 8 different languages. so that the common people can understand how they can make use of this facilities which are in CDFD. They don't have to pay anything, just the sample and the entire things are free of charge because DPT has sponsored this program. About 99 crore for 5 years. So this is for your information, and this was, the laboratory was inaugurated by former Vice President Magaya, and Secretary is the one who has launched the program at CDFD going on. And as I mentioned, if you come across any children, child with some of the rare disease, you can advise them to contact so that I close my talk by thanking most of my colleagues, Gladys Singh and others, and my collaborators. You can see that also part of some of these studies, and we also have large number of collaborators from outside India and also in the medical institutes, including AIIMS and so on. And research grants have been funded by many organizations. And thank you very much.
Sorry, he is supposed to be in the meeting at 11:30. We are very thankful to you, sir, because very systematically the things have been taken about human genome evolution, and then suddenly take certain examples, human diseases, how it is carried, particularly in the cases. But since students are not,
Yeah, so she wants to ask.
So you said you are going to do the prenatal counseling to check for the genetic disorders. So after doing prenatal counseling, is there any cure to prevent that disease?
Yes, so to do the prenatal diagnosis, if the fetus is carrying mutation, there's an option for termination of the fetus. Right? Otherwise, one can continue. Treatment is very difficult for any genetic disease. Of course, the CRISPR-Cas technology is there, but it's again it's not induced frequently. But the best option is avoiding the child with the mutations.
Thank you, sir.
Right here.
any other clarification questions, please? Students?
Sir, quick question. This is regarding the endometriosis. Now, as you said, in the last 2000 years, Indian populations are endogamous. How is this endogamy different from the endogamy that is seen in maybe African populations, which has been going on for a long time? Or you take, for example, Andamans. There also endogamy is there, but probably that endogamy started long time. So, what is the difference do you see among these endogamy? Does it have any difference in the diseases which they are there in these two kinds of endogamy that have started from different time intervals?
So endogamy, although Africans are very old population, they don't follow strict endogamy. Right? So Andamanis, although the population size is very small, they have to follow. But one possibility there, the individual with the disease doesn't survive, transfer the mutation to lactate. Unlike us, because there you can say those populations are naturally selected. Right? Whereas here, for example, somebody has a mutation. They get, for example, take the 25 days medication which does the cardiology. Somebody suffer from cardiac arrest, we immediately take them to hospital and make this person survive. and the mutation is transmitted to the next generation when the person marries. Whereas that kind of situation where they don't have any modern medical technology, they cannot survive, they cannot transmit the mutation to the next generation. Therefore, that's the end of the mutation. It can go generation after generation. Of course, it's transmit generation after generation. But only when the person marries, the other person carrying the mutation only has the children or child with the disease. Otherwise, it goes generation after generation. Heterozygous condition, there's no problem. Only when it exists in the homozygous condition, only then there's a disease.
So far, how many years have been identified?
The problem is, in a given point by time, if you go, there's no one surviving with the disease because children die at very early. So I mean, superficially somebody see the population health, they are all carrying the mutation in heterozygous condition. Unless two individuals with the heterozygous mutation marry, only then there's a disease. Otherwise, if somebody sees the population, we find compared to any other common disease, for example, cardiovascular disease, diabetes, this is quite common.
Any specific group, like as you mentioned?
Yeah, but purposely I did not mention the name of the population, but this is a group near Madurai, Tamil Nadu.
My second clarification is, I was also surprised with the people who are walking with 4 legs. Is there any further studies going on in that field?
That's a Turkish family. Probably there's some studies going on. This came out year back or 2 years back. That YouTube.
There are no scientific studies are
There is been done. One of the groups have been working on it. But I don't know whether something published there is. But all they work on these individuals. They might have identified the mutation. It's a crucial mutation helps in standing the students. So that's part of the.
What was your experience working with different population and how what was the response about the genetic disorders and the diagnosis?
Yeah, so those are different experiences. I can tell you two experiences. One is collecting samples from tribal populations of Andaman and Nicobar. We don't have any clue that who are we. There is a social worker who works with them closely, gives food material for them. They will take food material and then go to Jen. When we went, generally they were knocking at, we are aliens for them, right? So then the social worker conveyed message to them and they cooperated, there's no issue at all. Reaching that place is difficult. Okay, so we have to Port Blair, from Port Blair to a place called Bugon Creek. We ship, then some distance by road. Then we have to take a boat and go into the sea, make a U-turn, come to that on the side, on the bank, you are seeing a lot of crocodiles and so on. So that's kind of experience. The second experience is we also collected from other tribes, Chhattisgarh, when we went and collected samples, but they are not local areas. The Naxalites some of them. Of course, they didn't say anything to us. I was telling to the local person to wind up not go. The local person was saying, about many people are there to do the sample. He said, let's back up and go to other place. I told there are many still there. We collect and go. He said, no, sir, there are people waiting on the other side. While going, what he was saying that Naxalites are surrounded and asked us to leave. After we left, 1 week after we left, the place called Dandavada, there was a big bomb blast. Right. So that's collecting samples in that tribal. And I had another experience of collecting those 3 brothers who typically look like chimpanzee. Basic from Chennai, but they were in Bangalore in a circus company. This was 30 years back. So we made all attempt to go and collect samples. That Raj is saying who had some contact with the local DE and others. So with the help of that, I went there, but the circus owner did not allow us to collect the sample. Otherwise, you would have actually seen the link. So what actually made them to get back the chimpanzee? Somebody such experience.
Thank you, sir.
Hope you all enjoyed the lecture. Sir is actually MSc Zoology and studied in Madras University. After that PhD in Zoology. Then Sir has come to this high level status because of his dedication, hard work. I was working in his lab. When I go from 12 o'clock, he is working in the lab. Then when I come 9 o'clock, still he is working in the lab. Then I was telling Sir, when he is sleeping, I don't know. Always in the lab and working. That's why Sir has got this much awards and Royal and international notes and this. So this is just inspiration for you all. Please take and dedicate yourself and determine. Then nobody will stop to reach the high level. Okay? Being this, I really thank you so much, sir, for coming here in spite of your busy schedule. So, everybody tell thank you, sir. So, yeah. Thank you so much. And refreshment is there outside. Then we can have, and your certificate will be posted online. Okay? Thank you.